Good morning, dear students. So we have to start with the revision of the second chapter, which we have covered till today. That is the fundamental unit of life. So let's start with the revision. The term cell was coined by Robert Hooke in year 1665 in his book Micrographia after examining thin slices of cork under a self-designed primitive microscope. He had actually seen the dead walls of plant cells which had lost their living content and looked like small rooms. Anton von Leeuwenhoek described free cells for the first time in year 1674. Robert Brown in year 1831 discovered nucleus, the most important component of every cell. By this time, it had also been known that the cells were surrounded by some sort of limiting membrane. Schleiden and Schwann gave the cell theory. Accordingly, all plants and animals are composed of cells and cell in the basic unit of life. In 1855, Rudolf Virchow presented the idea that all living cells arise from pre-existing cells. This led to the modification of cell theory. Haeckel, in year 1866, reported that nucleus stores and transmits hereditary information. Discovery of electron microscope in 1940 made it possible for us to study complex structure of the cell and its organelles. All living organisms are composed of one or more cells, and accordingly, they are termed unicellular organisms. Example, amoeba, and multicellular organisms, example, plants and animals. In the former, a single cell constitutes a whole organism, while in the later, number of cells group together and acquire different functions to form various body parts. All multicellular organisms show division of labor, that is, different parts perform different functions. In fact, every living cell has got a division of labor. Its different cell organelles perform special functions and therefore constitute the basic building block, the cell. Thus, cell is the structural and functional unit of living organism. Cell differ in shape, size, and number. The shape and size of cells are related to the specific functions that the cells perform. However, number of cells is related to increase the size of organ or organism as a whole. Shape of the cell may be variable or fixed. Size of cells vary greatly. Most of eukaryotic cells are microscopic, ranging from 10 to 100 micrometer. However, few are large, size 2. Majority of the multicellular organisms are composed of numerous cells. Prokaryotic cells, on the other hand, are much smaller in size. Based on complexity of organization, cells are grouped as prokaryotic cells and eukaryotic cells. Prokaryotes are the most primitive organisms, example, bacteria, blue-green algae, or cyanobacteria, which lack nuclear membrane around their genetic material, which is called nucleoid. They also do not contain membrane-bound cell organelles in the cytoplasm. Ribosomes are, however, present. Eukaryotics have elaborate internal organization. Every eukaryotic cell contains a plasma membrane, membrane-bound nucleus containing the genetic material and other membrane-bound cell organelles in the cytoplasm. The organelles compartmentalize the cell to facilitate specific metabolic functions. Examples of eukaryotes are protists, fungi, plants, and animals. Each cell has three common features. Plasma membrane, nuclear material, may or may not be bounded by a membrane and a cytoplasm. All activities inside the cell, interactions of the cell with its environment are possible due to above three features. Viruses are the non-cellular organisms. They possess genetic material, DNA or RNA, enclosed in a protein coat. They do not show characteristics of life until they enter a living body to use its cell machinery to reproduce. A generalized eukaryotic cell has three main components: plasma membrane, nucleus, cytoplasm. Plasma membrane is the outer covering of the cell. It encloses cytoplasm and nucleus. Both cytoplasm and nucleus further have several components. Every prokaryotic cell and eukaryotic cell is surrounded by plasma membrane. Also, most cell organelles in eukaryotes are bounded by unit membranes. Plasma membrane is living, ultra-thin, elastic, and selectively permeable. Singer and Nicholson in year 1972 have proposed fluid mosaic model to explain its structure and functions. Accordingly, it is made up of phospholipid layer and two types of proteins, namely intrinsic proteins and extrinsic proteins floating about in the fluid phospholipid bilayer. Now in this line, children, you can see that plasma membrane is living, ultra thin, elastic, and selectively permeable. So it is very clear. That means it allows only selected material to come inside the cell. Clear? It is not semi. Semi means half. So it will be always selectively permeable only. Clear? Next is plasma membrane serves many important functions. It gives form to the cell, maintains its individuality, protects it from injury, keeps the cell contents in place. Being selectively permeable, it regulates the flow of selected material into and out of the cell. Its junction keep the cells together. Selective permeability of plasma membrane enables the cell to maintain homeostasis. Three types of mechanism, namely passive transport, active transport, and a bulk transport, are involved in the entry and exit of various materials through the plasma membrane. Passive transport of material is slow and does not expend energy. It occurs down the concentration gradient and does not use carrier proteins. Only small molecules pass through the plasma membrane by passive transport. Passive transport of materials occur by diffusion and osmosis. The process by which a substance uniformly spreads into another substance by random movement of its particle from a region of higher concentration to a region of lower concentration due to their kinetic energy is called diffusion. It is faster in gaseous phase than in liquid or solid phase. Ions, water, gases, etc. move across the membrane by diffusion. The diffusion of water or solvent through a semi-permeable membrane from a solution of lower concentration of solutes to a solution of higher concentration of solutes to which the membrane is relatively impermeable is called osmosis. Osmosis is of two types: endoosmosis, that is the entry of water molecule into the cells when surrounded by hypertonic solution, and exoosmosis, that is the exit of water molecules from the cell when surrounded by hypertonic solution. Active transport is rapid and requires use of energy in the form of ATP. It usually occurs against the concentration gradient and involves the use of carrier proteins. Glucose, amino acids, and some ions pass through the plasma membrane by active transport or co-transport. Bulk transport of materials involves special processes to transport macromolecules lipids, droplets, and solid food particles in large amounts across the plasma membrane by utilizing energy. These include endocytosis and exocytosis. The term endocytosis refers to invagination of a small region of plasma membrane, forming an intracellular membrane-bound vesicle. It is of two types: pinocytosis, that is non-specific intake of droplet of extracellular fluid through the cell membrane, and phagocytosis, that is the intake of solid particles by a cell through the cell membrane. Phagocytosis first puts out tiny protoplasmic processes called pseudopodia around the food, which later meet, fuse, and thus internal vacuole. The phagosome containing solid food is acquired by the cell. Endocytosis is not shown by plant cells because of their rigid cell wall and internal turgor pressure. Exocytosis is the process that involves fusion of membrane of the exocytotic vesicle with the plasma membrane to extrude its contents to the surrounding medium. It removes undigested food left in food vesicles. Secretion of substances, example, hormones, enzymes, and replacement of internalized membrane by the fusion of exocytotic vesicles with the plasma membrane. Plant cells, bacteria, and fungi have an additional thick porous coat outside the plasma membrane called the cell wall. It is chiefly composed of cellulose, that is polysaccharide. Adjacent plant cells and plant tissues are kept together by middle lamella, and they exchange materials through plasmodesmata. The process of shrinkage of protoplast of plant cells from the cell wall due to exosmosis when kept in hypertonic solution is called plasmolysis. Nucleus is the most conspicuous and largest organelle of the eukaryotic cells. It is generally centrally located and consists of double membrane nuclear envelope, nuclear sac, chromatin material, nucleolus, and nuclear matrix. It controls all the metabolic activities of the cell, contains genetic information, and is concerned with transmission of hereditary traits from one generation to another. Cytoplasm is a semi-fluid, jelly-like material having three components: cytosol, cell organelles, and cell inclusions. Cytosol is nearly transparent ground substance called cytoplasmic matrix or hyaloplasm. It contains about 90% of water, various cell organelles, and inclusions in it. The subcellular component of the cell are termed cell organelles or small organs. Except plasma membrane and the cell wall, all other organelles are found embedded in cytosol. These include endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, mitochondria, plastid, vacuoles, centrioles, cilia, and flagella, peroxisomes, etc. Next is endoplasmic reticulum. Is an extensive network of intracellular membrane-bound tubes and vesicles that occupies most of the cytoplasm. It is formed of cisternae, tubules, and vesicles. Rough ER has ribosomes attached on it on the cytoplasmic side. Smooth ER is without ribosomes. ER performs a variety of important functions, including packaging of materials, detoxification, formation of organelles, support, etc. Next is ribosomes. Ribosomes are the protein factories of the cell. On the basis of their size and sedimentation coefficient, they are of two types, that is 70S and 80S. Okay, the 70S and 80S we have not discussed, so need not to go into the detail. Chemically, these consist of rRNA and proteins and without a membrane around them. Golgi apparatus originates from RER that has lost its ribosome. It is generally located at a specific site close to nuclear envelope. It is membrane-bound, fluid-filled central sac of parallel, flattened, curving, arced, intercommunicating cisternae, and many peripheral tubules and vesicles. It plays important role in modifying, sorting, and packaging of materials coming from ER to synthesize in the Golgi itself.